Hey, how's it going, everyone? This is FixerMed. Welcome back to my high yield immunology review series for the USMLE Step 1 NBME CBSE and NBME CAS examinations. This will be part 4 of my multi-part video series covering the entire discipline of immunology in a comprehensive but high yield manner. So, without further ado, let's go ahead and get started on today's content. I'd like to open up today by talking about T-cell activation. So you might be wondering, what do I need to know about T-cell activation? So you need to know that the primary signal occurs when antigen-loaded cells interact with CD8 plus T cells, which are known as cytotoxic T cells, or CD4 plus T cells, which are known as helper T cells. And the primary signal involves the major histocompatibility complex binding to both TCR and CD8 molecule. MHC1 binds to TCR and CD8. MHC2 binds to TCR and CD4. Let's go ahead and go to the next slide. This is going to be a continuation of the first slide. Meanwhile, the secondary signal occurs where B71, known as CD80, or B72, known as CD86, on the nucleated cell or antigen presenting cell binds to the CD28 on the naive T cell, leading to T cell activation. So B71 or B72, CD80 or CD86 binds to CD28 in the secondary signal. Let's go ahead and move on to the next set of content. So now we're going to look at the outcomes of activation. CD8 plus cytotoxic cells directly eliminate antigen-loaded cells presenting on MHC1. CD4 plus T cells exhibit various effects based on differentiation occurring post-activation. So main takeaways here is primary signal, MHC binds to TCR/CD. Secondary signal, B7 protein binds to CD28. All right, now that we have that out of the way, let's go ahead and look at some review questions. During a lecture on immunology, a medical student is asked about the primary signal involved in T-cell activation. Which of the following statements accurately describes the primary signal required for the activation of CD8 plus and CD4 plus T cells? Alright, think I gave you guys enough time. If you need more time, please feel free to pause the video. Otherwise, moving on now. The primary signal for the activation of CD8 plus T cells, known as cytotoxic T cells, involves the binding of MHC1 molecules presenting antigen to the T cell receptor, TCR, and CD8 co-receptor. This interaction triggers the activation cascade necessary for CD8 plus T cell function. Similarly, for CD4 plus T cells, known as helper T cells, the primary signal occurs when the MHC2 molecules bind to TCR and CD4 co-receptor. Therefore, the correct answer here is MHC1 binds to TCR and CD8. Let's go ahead and look at what the other option choices were and why they're wrong. MHC2 binds to TCR and CD8. This option incorrectly pairs MHC2 with CD8, which is a characteristic of MHC1 interactions. MHC2 pairs with CD4, like we said in the earlier slides. MHC1 binds to TCR and CD4. This statement inaccurately associates MHC1 with CD4, whereas MHC1 primarily interacts with CD8. MHC2 binds to TCR and CD4. This choice correctly identifies the interaction between MHC2 and CD4, but incorrectly pairs MHC2 with CD8. CD4 binds to TCR and MHC1. This option confuses the roles of CD4 and CD8 in T-cell activation, as CD4 primarily interacts with MHC2, not MHC1. Let's go ahead and move on to the next question. During a medical lecture on T-cell activation, a student learns about the secondary signal required for T-cell activation. Which of the following accurately describes the secondary signal that leads to T cell activation? Alright, think I gave you guys enough time here. If you need more time, please feel free to pause the video. Otherwise, moving on now. The secondary signal for T cell activation occurs when B71, known as CD80, or B72, known as CD86, molecules on the nucleated cell or antigen presenting cells bind to CD28 on the naive T cell. This interaction provides the co-stimulatory signal necessary for T cell activation and proliferation. Therefore, the correct answer is E. CD80, CD86 binds to CD28. Let's go ahead and take a look at why the other options are incorrect. CD4 binds to MHC2. This option incorrectly describes interaction between CD4 and MHC2, which is part of the primary signal for T cell activation, not the secondary signal. CD8 binds to MHC1. This choice inaccurately pairs CD8 with MHC1, which is characteristic of the primary signal for CD8 plus T cell activation, not the secondary signal. CD80 binds to TCR. This statement erroneously suggests that CD80 binds directly to the T cell receptor, which is not part of the known signaling pathways involved in T cell activation. CD28 binds to MHC2. This option incorrectly identifies the binding partner of CD28, as CD28 primarily interacts with CD80, CD86, known as B71 and B72 molecules on the antigen presenting cell, not directly with MHC2. Let's go ahead and move on to the next question. During a seminar on immunology, a medical student is asked about the outcomes of T cell activation and the summary of the signal pathways involved. Which of the following accurately describes the activation outcomes in the summary of T cell activation? Alright, I think I gave you guys enough time here. If you need more time, please feel free to pause the video. Otherwise, moving on now. Upon activation, CD8 plus cytotoxic T cells recognize and directly eliminate antigen-loaded cells presenting on MHC1, which is their primary function. The secondary signal for T cell activation occurs when B71, CD80, or B72, CD86 molecules on the antigen-presenting cell bind to CD28 on the naive T cell, facilitating T cell activation and proliferation. Therefore, the correct answer is C. CD8 plus T cells directly eliminate antigen-loaded cells presenting on MHC1, and the secondary signal involves B7 protein binding to CD28. Let's go ahead and see why the other answer choices are incorrect. CD8 plus T cells release cytokines upon activation, and the primary signal involves B7 protein binding to CD28. This option incorrectly describes the activation outcomes of CD8 plus T cells and misrepresents the primary signal involved in T cell activation. CD4 plus T cells directly kill antigen-loaded cells presenting on MHC1, and the secondary signal is mediated by MHC binding to TCR-CD. This statement inaccurately attributes the function of CD8 plus T cells to CD4 plus T cells and reverses the roles of the primary and secondary signals in T cell activation. CD4 plus T cells differentiate into cytotoxic T cells upon activation, and the primary signal involves B7 protein binding to CD28. This choice inaccurately describes the differentiation process of CD4 plus T cell and misrepresents the primary signal in T cell activation. CD4 plus T cells release perforin and granzyme upon activation, and the primary signal is mediated by MHC binding to TCR-CD. This option misrepresents the activation outcomes of CD4 plus T cells and reverses the roles of the primary and secondary signals in T cell activation. That's all I have for the video today, guys. If you have any questions, comments, or concerns, please feel free to leave them in the comment section below. If you like this type of video, please feel free to leave a like and subscribe for more content like this. If not, this is FixerMed signing off. Be sure to have a good day and good luck studying, everyone. Goodbye.